Okay, hello everyone. My name is Rafa, and I will present in English because I'm still not used to speaking in French. But well, I am here today to try to explain to all of you what I'm going to do during this PhD project. It's a joint PhD program between us, the INRA, and the University of Leicester. And basically, what I will try to do is to optimize the aroma profile of Saccharomyces wine strains through hybridization techniques. So the first thing we have to have clear is the objectives. Currently, the main demand in the wine market is for new flavor profiles, for different molecules, improved flavor profiles. And that's why it's important. The first thing we will try to do is to provide a better understanding of how this complex trait, the aroma profile, is regulated at a genetic level. That is the first objective of the thesis. And as a consequence, we have a second objective, that is the industrial objective, in collaboration with L'Allemand. And we will aim to generate novel hybrids, interspecific hybrids, that is between different species inside Saccharomyces, trying to optimize the aroma profile and other interesting traits for the wine industry. So, for this purpose, the experimental design can be divided in 4 steps. I'm now in this first step, that is the screening of the candidates to be parental strains in the hybridization. Then, once we have selected the best strains, I will perform the genetic analysis, the QTL mapping, and also metabolic flux analysis to correlate the central carbon metabolism with the nitrogen metabolism and to see how that influences the aroma production. Once we have this genetic information, we will use it to create novel hybrids, as I told you before. But in the food sector, we are a bit limited because we cannot use techniques that imply the use of genetically modified organisms. I will explain a bit more of that later. And the last step, hopefully, once we obtain the hybrids, will be the industrial implementation, that is the scaling up and so on. So basically, this is the scheme of what I'm going to do. And now, as I told you, I am in this first stage, that is the screening of the candidates to be parental strains. All of the candidates are inside the genus Saccharomyces. Some of them are pure species, and others are already hybrids. So maybe we will have the opportunity to combine a hybrid with another species to make a hybrid between 3 species. This is one of the possibilities. The initial point is 90 candidate strains, some of them from here, others from the University of Leicester. And I will do the selection mainly in basis of 2 parameters, 2 criteria. One of them is the fermentation capacity of the strains. I am now at this point analyzing how the different strains behave in wine-making conditions. And the second most interesting trait that we want to optimize is the production of interesting aromas. For that purpose, I'm taking samples of the fermentations at different points to be able to measure by GCMS the main aroma metabolites. And I will also measure by HPLC some other important compounds for the fermentation metabolites that are consumed or produced during the fermentation. And well, there is another trait, another phenotype that is quite interesting under the point of view of the aroma production. That is the cryotolerance. This is very important because if we do all the process of the fermentation at a lower temperature, we will be able to retain the most part of the aromas because they are volatile molecules and they will evaporate if we use higher temperatures. As I told you, I already have some results regarding the fermentation kinetics. Basically, this is the setup. As I told you, we have 90 strains. I'm using 2 different temperatures, 16 and 22 degrees, and making 2 replicates of each one. This makes a total of 360 fermentations in a volume of 250 milliliters. These are the first results of manual fermentation, that is waiting manually. And here you can see some results of fermentations using the robot. As you can see, this fermentation is not yet finished, but these are simply two examples to let you see the difference between performance of different strains, different candidate strains. Here we can see clearly which one is the best performing strain, but to be able to do a selection in the basis of quantitative data, we will extract, using the R software, we will extract some kinetic parameters like the Vmax and the time that gets to overcome the lag phase and so on, to be able to compare quantitatively and select the best parental strains. Once we have selected the parental strains, on the basis of all of these criteria, we can start with the QTL mapping. The first thing we have to understand here is that aroma production is a complex trait. This means that the production of each metabolite depends on the influence of several genes. So that's why we have to use QTL mapping to detect regions in the genome that influence the production of this metabolite. These regions in the genome, this loci, is what we call QTL, or quantitative trait loci. To be able to perform this technique, we need two parental strains. At this point, we will have already the parental strains. And the procedure is relatively simple. We cross them, we make them sporulate, and at this point, we can analyze this population or make another run, another generation. We make them cross, sporulate, and we will make a third generation, a fourth generation. As more generations we do, the fragments that the daughter cells will take from the parent strains will be shorter, and this increases the resolution of QTL mapping. Anyway, at some point we stop, in this case in the F2, with 2 generations is enough, and we start to analyze this population of segregants. We will get 2 types of data. First of all, genotypic data. This is basically you sequence the whole genome and you analyze the sequence looking for different genetic markers, different polymorphisms, normally microsatellites or SNPs. And on the other hand, phenotypic information. This is basically the measurement of all the traits or the parameters that I told you before, the fermentation capacity, the aroma production, and all the criteria I told you. Once we have these 2 types of data, we can combine them and make a linkage analysis to detect or to map the QTLs. And the QTL mapping analysis will be done. But the final objective of all of this is to identify specific genes with specific functions. So once we have the QTLs, we can look into the QTL, we have the sequence, and we can go to a database and say, okay, this gene maybe has this function because it's implied in this metabolic pathway. Okay, so with QTL mapping, we obtain all this useful information that will be useful in order to create the hybrids. This is the final step, the final research step. And well, the first thing we have to understand here is that interspecific hybrids are already being used in industry. Probably you know Saccharomyces pastorianus. This is an interspecific hybrid that was created spontaneously during lager beer fermentation, and it's a combination between Saccharomyces cerevisiae and eubayanus. This strain, this species, was able to combine positive effects from the different strains. In this case, she took the fermentation capacity and flocculation ability of cerevisiae and the sugar utilization and cryotolerance from the eubayanus strain. In addition to this, normally when we do hybridization, we have an effect, we can have an effect that is called heterosis. This means basically that sometimes the the phenotype of the daughter cells will be better than the one that we have in the parental strains. So, bearing this in mind, we will try to do the same, or at least we will try to obtain the same result, but combining the aroma profile, good aroma molecules that are positive for the wine, with the cryo tolerance of another strain or the fermentation capacity of another strain. At this point, it's good to say that we don't want to increase all the aromas in the wine. Some of them are positive, but others are negative. They are called off flavors. And by doing hybridization, it has been shown in the literature that you can increase the concentration of some compounds and decrease at the same time the concentration of non-useful compounds. There are several techniques to make the hybridization, but as I told you before, in the food sector we are a bit limited because we cannot construct GMOs. And for some of these methods to do hybridization, at some point you introduce a gene, for example a selection marker, in order to select the hybrids. I don't know yet what technique I will use, but I will go during November to Leicester to discuss with my supervisor. He's the expert on this, so he will tell me what techniques I will use. The only thing I know is I will use two different techniques. One for the QTL mapping, and the other one, mass mating probably, for the creation of the industrial hybrids, because you don't necessarily have to modify intentionally the genes. And well, this is basically everything, so thanks for your attention.